one of the pioneers in nanotechnology, not only in Spain but also in Europe. So the agenda for today is a bit of introduction about vaccination and adjuvants, and then I'm going to focus on mucosal vaccines and especially in nasal vaccines. So a vaccine typically contains an agent that resembles a pathogen and that stimulates the body and to keep a record of it, and then in the future when this pathogen reaches the body has the weapons to fight against it. So I have highlighted the word agent because this maybe makes the major differences between the classical vaccines and the modern vaccines. Classic vaccines, since the 18th century, when Edward Jenner started to work with smallpox, mainly consisted in life-attenuated microorganisms or inactivated pathogens. And they generated quite good immune responses. They didn't need adjuvants, but they were quite unstable and they could revert to the pathogenic form. We have seen along this Congress that modern vaccines are more based in recombinant proteins, peptides, and genetic material. They are much safer, easier, and cheaper, but they are poorly immunogenic because they don't resemble very much the pathogen of the origin. So with these modern antigens that are much safer, we need to use different adjuvants. And the main categories of adjuvants, we have both immunostimulatory molecules and delivery systems. In the last decades, we have discovered that toll-like receptors play an important role in the origin of the immune response, but we have also toxins, saponins, cytokines, and macromolecules. And in this case, I have highlighted polysaccharides because we have worked quite a lot with them, like chitosan, dextran, or glucans. They are recognized by antigen-presenting cells, and they are usually biocompatible and biodegradable, and they can be used for preparing micro and nanoparticles. And this delivery resistance has adjuvant properties because in some way we can simulate a pathogen because they have similar sizes. So the body finds them quite exogenous, so the body reacts against them. And we can even encapsulate these other molecules in the nano or micro systems, and they are quite useful for mucosal administration. These are the adjuvants that have been approved for human uses in vaccines. The reference is still aluminum salts, but in the last decade, some emulsions and liposomes have been used for new vaccines. There is quite a lot of research in this field, especially with cancer vaccines. So focusing now on mucosal vaccines, why are interesting mucosal vaccines? For several reasons. First of all, mucosa are the main entrance of several diseases. For example, influenza in respiratory tract or HIV in genital tract. So it's important to have a good immune response in that part of the body. It's the first line of protection against diseases. And mucosal vaccines can generate both systemic and mucosal protections, IgG and IgA. And parenteral vaccines mainly generate systemic response. So it's good to have mucosal response for a lot of illnesses. And then mucosal vaccines are especially suitable for needle-free vaccination. And this is important not only because almost anyone likes needles, but it's useful for mass vaccination with epidemic outbreaks. So especially important in developing countries. And the other advantage of mucosal vaccines is that they are much easier of production. We can think in oral vaccines, they need much less requirements of sterility and purity like a parenteral vaccine. So a bit of immunology. mucosas that are present along the body. They are in contact with environment, so they possess large surface, especially intestines and lungs. All the mucosa have under the epithelium this mucosa-associated lymphoid tissue, and they are very rich in immune cells. So we can generate there a quite important immune response, not only locally, as I said, but also systemically. And we have some inductive places and some effector places. For example, intranasally, you can generate a very good response in respiratory tracts, in genital tract, and a bit less important in gastrointestinal. So the main challenges for mucosal vaccination are the enzymes that are present in the mucosa, that they can degrade the antigens, the barriers that we have to overcome, especially the mucus barrier and the epithelium barrier. We have to release the antigen in the proper site, and we have to stimulate in some way the body to tell that this protein, this peptide, they must react against it. And nanotechnology can offer solution to most of these problems. They can protect the antigen inside a nanostructure. They can help the penetration of the different barriers, the mucus barriers, the epithelium. They can provoke a sustained release of the antigen. And apart from their own adjuvant effect, we can co-encapsulate other adjuvants. There have been a lot of different nanosystems employed for vaccination. We can classify them in polymeric, lipidic, and pathogen-derived. We have micro nanoparticles, we have liposomes, nanoemulsions, virus-like particles. And the main compounds that have been employed to prepare these nanosystems are synthetic, the polyesters. We have seen the problems with polyesters and the acidic environment after hydrolysis, and natural polymers, and here the leader is chitosan. Sorry. So I'm going to focus now in the nasal administration of vaccines. The nasal cavity has some advantages. It's a large area. Comparing with the gastrointestinal tract, the reduced enzymatic activity. The epithelium is quite permeable. It's easy to administer. And in this case, nanoparticles can increase the residence time of the antigen. If we use compounds that are mucoadhesive, we can prolong the residence time. We can help the penetration of the antigens in the epithelium. This was one of the first results in the group of Maria Jose. They started to work with tetanus toxoid, and they proved that the nanoparticles could cross the epithelium, and they established a relation between the size of these polylactic acid nanoparticles and the efficacy in the penetration. So nanoparticles could efficiently cross the epithelium, but microparticles are a bit more hindered. Then, as we saw in previous talks, pegylation has an important role here in improving stability of nanoparticles in the nasal cavity and in helping with the penetration of the antigen to the epithelium. Then, when we started to realize that the polyester has some problems with the activity of the antigens, we moved to more friendly materials like chitosan. In the group of the Maria Jose, they were the first of reporting chitosan TPP nanoparticles by ionic gelation technique these nanoparticles have been employed in different fields of nanotechnology. Then a big step happened when 10 years ago we were funded by Bill and Melinda Gates Foundation to create a single-dose vaccine and needle-free delivery systems that are quite important in the developing countries. So we moved to different nanostructures, especially this one, the nanocapsules. They have an oily core. We can put here immunostimulant oils like esqualene or vitamin E. We have stabilizing this oil, different surfactants, and then it's important this polymeric shell that we can have here different polysaccharides or polyamino acids. And we used as antigens, reference antigens, hepatitis B antigen and influenza antigen. So I'm going to show now some results with hepatitis B antigen and chitosan nanocapsules that contain in the nucleus imiquimod, that is a toll-like receptor. And this system fulfilled expectations for nanovaccines in the sense of adjuvant effect. We can have a single-dose vaccines, needle-free, and they are quite thermostable. So they are perfectly internalized by macrophages, and this is quite common. In this size, they are in the range of 200 nanometers. Then if we compare intranasal administration of these nanocapsules with intramuscular administration using alum as adjuvant, we can have similar protective levels. Of course, the dose for intranasal vaccination needs to be superior, but anyway, even with 10 times more antigen intranasally, it's quite very good results to have similar protective levels. Then one of the advantages of using nanoparticles is that we can balance the humoral and cellular response. So alum is mainly humoral responder. So with these nanoparticles, we can have a much more balanced IgG1, IgG2 response. And we have proof that all the components, the chitosan shell and the imiquimod, they are essential for having the best ratios of antibody responses. This is intramuscularly, but it's interesting also that with a single dose of these nanocapsules, we can have quite good levels of antibodies comparing with 2 doses of nanocapsules intramuscularly and with alum. And if we compare them, they are in similar levels. Then these nanocapsules can be freeze-dried with different cryoprotectants. We have a solid that can be stored at room temperature, and the antigen maintains with integrity along at least one year. We can see that we have the antigen inside the nanocapsules instead in solution, or we have a quite loss of the antigen. To finish, I will tell you some projects that we are involved now. We are working in an HIV vaccine. We collaborate with University of Manitoba in Canada. We are funded by NIH in the States. And here we have 2 different ways to tackle with HIV virus. People in the University of Manitoba identified 2 proteins that could be effective targetings for vaccination. There are envelope proteins that are very difficult to isolate, so they proposed us to use the plasmids. So our role here was to encapsulate the plasmid codifying for those 2 proteins in nanoparticles, and then using these nanoparticles for intramuscular administration to achieve the desired results. And this in vivo is ongoing, so I cannot show you results. We have some results in the other strategy. In this strategy, people in Manitoba identified 12 different peptides belonging to a single protease that is essential for virus replication. And these 12 peptides we encapsulated in chitosan nanoparticles, in different chitosan nanoparticles, and then we use a cocktail of the 12 different peptides for boosting macaques intranasally. And after the boosting with these nanoparticles, we have seen an increase in the response of the macaques. And even some of them could be challenged 5 times without suffering the illnesses. It's quite important, this formulation, so we get involved in other European projects, the NanoPilot. The main objective of this project is to scale up a formulation, in this case the chitosan nanoparticles with 1 peptide, using microfluidics to have a continuous way of preparation the nanoparticles, using GMP materials and production with high advanced characterization techniques. And this is all. I would like to thank Professor Maria José Alonso and all the people involved in the vaccine part in the University of Santiago de Compostela. Thank you very much.
Thank you, Jose. Are there any questions from the floor? Well, I have one. That was really nice. Whenever you administer whatever formulation you have nasally, is there any issue? I mean, is it a liquid that you put inside the nose, or is there any issue with loss? You know.
Well, you have to take care the device especially, and the animal model is also important because, for example, rats are not very happy having something administered intranasally, but usually we usually administer with syringe very slowly and maybe in several times, and you can use, well, actually nanocapsules are usually a bit viscous.
Okay, they're viscous. Right.
Yeah, viscous, and chitosan makes them quite mucoadhesive. So,
okay.
Yeah, generally.
And is that the sort of formulation you used in the macaques then, the viscous type of formulation? Well,
in macaques we use nanoparticles. They are quite concentrated, so there are some viscosity also.
Okay, all right.